So today we are going to study about the first chapter in microbiology, and this is the first lecture of it. Mainly we are going to study about the means introduction and classification of microbes. So first let us know the definition of what is microbiology. Definition of it. So microbiology, it is the study of microorganisms which we cannot see with our naked eyes. It's easily understood, means the microorganisms which we cannot see with our naked eyes, we study them under microscope, and this study is called the study of microbiology. Means this is the broad definition of it. So this is the important point. Majority of them are harmless, means majority of these microbes are harmless. For example, saprophytes and common cells. Under saprophytes we get mushrooms. Okay, the mushrooms we eat or normally grow, they are harmless. And common cells, this, as we have studied during your need days, commensalism is a good example. Here the microbes that are there, they themselves gain, but they do not cause any harm to the body. Means the relation here is plus 0, plus 0. This is the relation. They do not cause any harm to your body. As you can see, commensalism is a symbiotic relationship where one party benefits while the other is not affected. This can be written as plus 0. Like in this example, you can clearly see this person is taking shade under this larger animal, but he is getting advantage of the shade, but he is not causing any damage to the larger organism. So this is plus 0. Like this, many bacteria stay in our gut. They take shelter in our gut, they get a place to live, but they do not cause any harm to our body. So that is a type of commensalism. They do not cause any harm. So two types of microbes, saprophytes, mushrooms come under them, and commensals. And now mainly in MBBS or in medical field we study about the pathogenic microbes. Pathogens, these are the microbes that cause diseases in us. Microbes that cause disease in our body are known as pathogens. We mainly study pathogenic microbes which infect and cause diseases in humans. So this is the main funda of studying clinical microbiology in MBBS. Now let us move on. So what is probiotics? Probiotics, they are good microbes that promote good health in us. Promote good health in us. They are good microbes which promote good health in us. And the substances that promote the growth of probiotics is called prebiotics. So it can be remembered quite easily. Probiotics, they promote good health in us, and prebiotics are substances that prebiotics promote the growth of probiotics. So here are some examples of probiotic foods and prebiotic foods. These prebiotic foods, they promote growth of probiotics. They act as a catalyst. Yes, growth of probiotics. So as you can see, like asparagus, leeks, onion, garlic, banana, berries, oats, legume seeds. These are prebiotic foods. They will promote the growth of probiotics. And probiotics are yogurt, kefir, kimchi, miso, and fermented veggies. A very good example of it is pickles. Yes, we also call it as achar, but not the paratha. Example of it is Lactobacillus rhamnosus. Okay, this is one example. Lactobacillus rhamnosus. They, what is their function? They decrease the number of cases of diarrhea in younger kids. They decrease the amount of diarrhea, the cases of diarrhea in younger kids. This is Lactobacillus rhamnosus. And another example of it is Saccharomyces boulardii. Saccharomyces boulardii, they decrease the risk of antibiotic associated diarrhea. Saccharomyces boulardii decrease the risk of antibiotic associated diarrhea. So two examples we learned. One is Lactobacillus rhamnosus and another is Saccharomyces boulardii. This Lactobacillus rhamnosus, they decrease the diarrhea in young kids, and this Saccharomyces bovedi, they decrease the risk of antibiotic associated diarrhea. Now we are studying the advantages of probiotics. So as we know, the main advantage is it promotes good health in our body. So apart from it, means how it does, they prevent binding of pathogens to host cell receptors. Suppose this is a pathogen and this is the host cell, and here is the receptor for it to bind, but this will prevent its binding. Yes, this prevent binding of pathogens to host cell receptors, these probiotics. So like this way they are promoting good health. Let us see the second example. So they enhance immune response, they enhance the response of our immune system, probiotics. Another one is they decrease inflammatory reaction against pathogens. So they help in decreasing the inflammatory reactions that happen when a pathogen reacts with our body. So these are the advantages. Now we are classifying microbes. Okay, how we will classify the various kinds of microbes? First, let's go under viruses. Now as we all have studied, the viruses are non-living, not non-living or living, means they are neither of those. They are neither of those. They are non-living. Okay, mainly they are non-living, but they get activated when they enter the host cell. So we neither classify them as living nor as non-living. Okay, so here you can see broadly some examples: bacteriophage DNA, tobacco mosaic virus TMV, it has RNA, adenovirus, influenza virus, the latest was coronavirus. Yes. Now see, as I told, they are neither living nor non-living. They are non-living when they are present in surroundings or in substances. They are non-living, but when they enter their host, they become living. So they are obligate intracellular infectious particles. Okay, they are obligate intracellular infectious particles. The now let us know what is the smallest. Smallest is the parvovirus and largest is the pox virus, which causes smallpox. Parvovirus is 20 nanometers and pox virus is 200 nanometers. Now viruses are measured in nanometer. This is their unit. You can see nanometer. Okay, this is the unit of the size in which it is measured. Viruses. Parvovirus is the smallest and this pox virus is the largest. PP. PP. P for parvo, P for pox. And pox only has 3 letters. 3 letters, so it is the largest. Means small. Letters are small, 3 letters small, so it is largest. And parvo it has 5. As the letters are big, it is the smallest. This is a small, means a normal trick you can use. Not a trick, just a way to remember. They have either DNA or RNA. This is very important point. What we generally have a misconception that a virus can have DNA or RNA or both, but no, the main thing is a virus only has a DNA or only RNA. It cannot have both. DNA is double stranded like this and RNA is only single stranded. They multiply by viral infective cycle. This is the way they multiply, viruses. Viral infective cycle, the way they multiply. Okay, now virus is done, just a normal classification. Now let us go to bacteria. They have both DNA and RNA. This is very important. They have both DNA and RNA. Both DNA and RNA. Like in viruses, only RNA or DNA, but in bacteria they have both the DNA and RNA. Replication is by binary fission, means one it goes divides to 2 and then divides to 4 like this: 1 2 4 8 16 32 like this. The normal formula for it is 2 to the power n, where n belongs to 0 1 2 like this and so on, like this is the normal formula. Binary fission, 1 divides to 2. Okay, so you can remember this formula also. What happened, this formula or like this: one bacteria divides to 2, then further this to into 2, then again this into again, like this. Important thing is this bacteria are prokaryotes. Prokaryotes means the organism which do not have a well defined nuclear membrane, means they are in nucleosome, all are placed haphazardly. Okay, so they are placed haphazardly. Prokaryotes. You can also remember these prokaryotes by primitive, like they are the most primitive, so they are prokaryotes. Not well defined cell structure. Now see, they are single circular double stranded DNA molecule. This is the genome. Understand it very nicely. This is for bacteria. They are single circular double stranded DNA molecule. Single, okay, let me do. Single means only there will be one DNA, and this is circular. So this will be circular like this. We will complete the circle, okay? Circular double stranded. Okay, this is double stranded only, double stranded DNA. This is the genome mainly. As I have drawn it here, single double stranded, two strands are there, and circular. This is for bacteria. The space occupied by this genome in cytoplasm is called nucleoid. Okay, suppose this is the entire cytoplasm. The space occupied by the genome inside the cytoplasm is called nucleoid, this portion. Now again classification, the smallest and the largest. The smallest is mycoplasma, mycoplasma, and the largest is bacillus anthracis, bacillus anthracis. Okay, you can see it here, and their unit, they are measured in micrometer. Okay, micrometer. This is 0.21 to 0.3 micrometer, and this is 4 to 8 micrometer. They are unicellular, single cell. This is very important. They are unicellular. They have cell wall made up of peptidoglycan, murin, mucopeptide, muramic acid cell wall. Just remember this, this will be beneficial. Peptidoglycan. Their cell wall is made up of peptidoglycan, okay? Can you see how we are transitioning from dead means either living or non-living, living or non-living, to living, then living, then this was dead, this is prokaryote, then we will go to eukaryote like this. So, but this is, and this is prokaryote and single celled, and they are made up of peptidoglycan. Remember this bus. And ribosomes, as we know, they are prokaryotes and they have ribosomes of 70s. 70s is formed of 30s and 50s. It is found of 30s and 50s. You can clearly see this is the 70s of prokaryotic ribosome. This is the 30s subunit, the smaller one here, which is topped up by bigger one, the 50s. It has 5s RNA, 23s RNA, and 16s RNA in the 30s subunit, and these two together form 70s. Now see, this S is very important. As you can clearly see, what does S mean? This is very crucial. First, learn the meaning of S. What does S stand for? S stands for sedimentation coefficient or Swedberg constant. Sedimentation coefficient or Swedberg constant. S stands for. Now, what does it mean? It is the velocity with which the ribosomes settle down after centrifugation. Suppose we take this bacteria, this ribosome. Now we put them in a test tube and rotate it, means do the centrifugation process. After centrifugation process, it will settle down, it will settle down like this. Now the speed at which, the velocity at which it settles down is known as Swedberg constant or sedimentation coefficient. With the name, it is clearly, you can clearly understand by the name, it is Swedberg sedimentation coefficient. Okay, so this is the S. Now let's move on to another, fungi and protozoa. Fungi and protozoa. Fungi denote as F, protozoa as P. Now see, we are going to more advanced stages. Now they are eukaryotes. Okay, from prokaryotes we moved on to eukaryotes. And eukaryotes, you can remember, eukaryotes are those which have well defined nuclear membrane. Means they have good mitochondria, good cells, good ribosomes. This is it. Okay, eukaryotes, they have multiple linear chromosomes. Now see, size, fungi are little bit smaller and protozoa are bigger. It is 5 to 10 micrometer and this is 10 to 20 micrometer. And now here are some facts or some points which you can note. Protozoa, as you can see, 3 O's are there in the spelling itself, so it can be signified as 0 and it can be again translated as they are unicellular. This is just a trick to remember. Protozoa, they are unicellular. How unicellular? As the name has 3 zeros. Just a way to remember it. They are unicellular. Now see, fungi may be unicellular or multicellular. Fungi can be both, but protozoa only unicellular. And fungi, the unicellular fungi is yeast. Yeast. You already might have studied fungi. They are non-motile. Fungi, they are non-motile. Means they will not move. They will stay fixed. Okay, very important. Fungi, they will not move. Fungi remains fixed. And protozoa motile. Exception is BPT. Remember this trick. BPT. BPT stands for Babesia plasmodium and Toxoplasma gondii. BPT. So fungi is non-motile, but protozoa is motile. Exception is BPT. These all are non-motile. These are very important. This may come as MCQ. Now F and P means fungi and protozoa have both DNA and RNA present in nucleus. So as we are advancing, they both have DNA and RNA. F and P. Yes. Now they divide sexually by meiosis. Meiosis, you learned one cell. If it has 2n, they will divide into 2 cells which will have n and n. So this is the normal funda of meiosis. Let me just label it. Suppose this is 2n. This will divide into 2, n and n. This is meiosis, and asexually by mitosis. Mitosis is similar only like cell growth and repair. They will divide into 2, each having 2n, 2n, like this. Growth. Now ribosomes. Their ribosomes is 8ts, made of 40s and 60s. Yes, I already told Swedberg unit, and like this. 40s and 60s combined to form 8ts. I think you can, the way you can remember is, in ribosome case all are even. Suppose in 70s, how it is formed? It is formed by 50 plus 30, means 50 and 30. So see, all are odd numbers, 7, 5, 3, and in case of 80, it is formed by 40 and 60. See, again even numbers. So like this you can easily remember it. 70, 3 and 5, and 60, sorry, 80, it's formed by 4 and 6. All are odd numbers. So like this way you can remember it. So done. This is also completed. Now see, fungi cell wall is made up of chitin. This is important, chitin. Now, what is chitin? Chitin is nothing but it is a homopolymer of N-acetyl glucosamine. N-acetyl glucosamine in short form NAGA, means it is like this. One unit of NAGA, then another unit of NAGA, then another unit of NAGA, then another unit of NAGA, like this it happens. Another unit of NAGA. Homopolymer means it only contains N-acetyl glucosamine. These are just basic stuff which we have learned earlier. Now see, F for fungi. Yes, it's fungi only, not something else. F has a unique sterol in their cell membrane. This is ergosterol. This is a very important point. This is new to you all. Fungi have unique sterol in their cell membrane, that is ergosterol. Just remember it. Ergosterol is present in fungi. Like you can F after, means before F what comes? E. So E for ergosterol, E present in F. Like this you can remember. E present in F. Antifungals that act on ergosterol are A and I. A is amphotericin and I is imidazoles. A and I. Like this is a trick. EAI. EAI means what we know, the antifungals that we use are mainly used for killing the fungus. So what, now specifically there are some things, some antifungals that act on ergosterol, the sterol that is present on cell membrane of fungi. So which are those antifungals that act on ergosterol? This is A and I. A stands for amphotericin and I stands for imidazoles. Okay, EAI. Like this you can remember, EAI. Now see, this is a question. Okay, name the parasite that has ergosterol. This is Leishmania donovani. It causes leishmaniasis. This is important. This is a parasite. Parasite means, again you might have learned plus and minus. Means what does this signify? Means if they remain in your body, they will harm you. They will harm you, but they will themselves gain advantage. So plus and minus. Commensalism, it was plus and zero. They are not harming, but parasites they harm you. So plus and minus. And this is Leishmania donovani. They cause leishmaniasis. This parasite has ergosterol. Okay. Now they are helminths. Now we are moving on. They are eukaryotes. Now they are bigger in size. They are part of animal kingdom. Okay. They are bigger in size. Adults can be seen by our eye. But why we are studying them in microbiology? Because we are not studying them, we study their egg and larvae form, because they need microscopes to be seen. Their egg and larvae form of helminths require microscopes to be seen, and they are not microbes. Now there are two types of unconventional infectious agents. All have been finished. The two types of unconventional means uncommon, not common. They are viroids and prions. Now see, what does the name suggest? Prions means proteins. P for prions, P for proteins. And viroids, they do not have proteins. Let's see. Now see, viroids. They are made up of RNA, okay, single stranded, okay, single stranded, covalently closed, okay, molecules with internal base pairings, like this, but they are covalently with internal base pairing, means they have base pairing within themselves, like G triple bond with C and A double bond with T. Is it correct? G triple bond with C and A double bond with T. Is it correct? They bond with themselves within themselves. Is it correct? No, it is not correct, because see, it is RNA. RNA doesn't have thymine. It has, instead of thymine, it has uracil. So this is very important. Don't forget it. If it was DNA, then it would have been thymine. But in case of RNA, G will triple bond with C, but A will double bond with uracil. Okay, so remember it. Just few points. Proteins absent. Proteins absent in viroids. Plant pathogens, they mainly affect plants, and no human diseases. They cause no human diseases. Sensitive to nuclease. Why they are sensitive to nuclease? As they only have nucleic acid in them. Okay, they do not have the protein part, only have the RNA part. This RNA part. So they are sensitive to nucleases, as they are made up of nucleic acid. Nucleases break down nucleic acid. We all know. Okay. Now move on to prion. Prion, these are proteinaceous infectious particles. Okay, means they only have proteins. They do not have the nucleic acid in them, only the proteins. They don't have nucleic acid. Okay. Disease in humans, which the cause is spongiform encephalopathy and prion diseases. They were discovered by Sir Stanley Prusner. They were discovered by Sir Stanley Prusner. Now this is very important point. They cause no immune response. This is very dangerous. They will cause no immune response but will damage the body. No immune response, because they are proteins of our own body only, but they become misfolded. The proteins present in our body which get misfolded lead to the formation of prions, and they do not even give immune response. So we cannot also understand. So they are very difficult to inactivate. It is a very dangerous situation for prions. Okay, very dangerous. Now, some examples between prokaryotes and eukaryotes. Just the normal beginning. Prokaryotes are primitive and eukaryotes are modern. Okay. So see, prokaryotes, they have only like this nuclei, no nucleus, no nucleus, means not well defined nuclear membrane, not well defined nuclear membrane. But in case of eukaryotes, you can see the nucleus, and inside it there is nucleolus, and nucleus is the site for RNA synthesis. So they are well defined. Well defined. We all know this from Chotowala. Now see, here is no histones. Histones are present. Histones are basic proteins like H1, H2, like this. Histone octamer. DNA winds around the histone octamer. That histones. No introns, only one exons. Only one exon. No intron. But here in eukaryotes it has a interesting feature, like intron, exon, intron, exon, intron, exon, intron, exon, like this. Genes are discontinuous. See, discontinuous. Introns are not as expressed, exons are expressed. So suppose this. Suppose like this. Yes. So this is exons, and this is introns. Okay. You might have learned earlier that we splice of these segments. These do not code for anything. Okay, these are not expressed. But these exons, these are expressed. By the name only you can see exon expressed. Okay, they are expressed. They are code for something. Okay, like little bit mature. But in case for what? In case for prokaryotes, only one exon. Extra chromosomal genetic material present is plasmid. This is very important. Plasmid. What is plasmid? This is extra chromosomal genetic material present. Like see, the genetic material that is present outside the chromosome, that is extra genetic material. Like here, outside, in circular form. These are plasmid. Outside the chromosome. They are mainly circular. Plasmid. And so let's continue. And here extra chromosomal genetic material is mitochondrial DNA. Mitochondria, we know the powerhouse of cell, they itself have their own DNA. So mitochondrial DNA, the extra chromosomal material, here is plasmid and here is mitochondrial DNA. But this plasmid is very important, we will read about it later only. It is single circular double stranded DNA. See, single circular double stranded. Double stranded like this means two strands will overlap and circular. Okay, like this. Single stranded double circular. It is inherited from maternal cell. From mothers it comes, maternal cell. Here are few points about bacterial chromosomes. Bacterial chromosome has approximately 3000 genes. Okay, 3000 genes. Around 110 genes are present in plasmid. This is very important. 3000 genes total. Out of them 1/10. 1/10 means 300. They are present in plasmid. Plasmid is also single circular double stranded DNA, except in Borrelia they have linear plasmid. As I told you, they are single circular double stranded like this, but in Borrelia they are linear plasmids. Plasmids are not essential for life of bacterium. For bacteria means prokaryotes, mainly present in prokaryotes, they are not important for life, means like for our body, heart, liver, kidney, brain, these are all essential. Without this we cannot live. But plasmids are not essential, but they are beneficial. They are present for betterment of a bacteria, but they are not essential. Without plasmid a bacteria can live, but if there is plasmid it will live more nicely, it will have a better life. Therefore they are not present in all bacteria. As I know, it is not essential, so they do not need to be present in all bacteria. Plasmids are autonomous self-replicating structure. They automatically replicate within themselves. So what we learned important is total there are 3000 genes. Out of them 300 means 1/10 are present in plasmids. Plasmids are mainly circular, but in Borrelia it is linear. In Borrelia it is linear, and they are self-replicating. This is a very important point. Self-replicating, and another important point, they are not essential. Now functions of plasmid. Functions of plasmid, it can be remembered easily by the name itself. Plasmid, plasmid. So 7 points we will learn. So for P, they are pili, which are organ for attachment. We will read about it later. Organ of attachment. Pili helps for attachment. L stands for light UV resistant DNA repair enzymes. Means UV resistant DNA repair enzymes. UV light resistant DNA repair enzymes. For A, antibiotic resistant enzymes. They will resist antibiotic. For S, They secrete endotoxins. For A, metal resistance reductases means resistance to some metals like mercury and silver. For mercury, they are mercurochrome and merthiolate. And for I, just add a N beside it, nitrogen fixation enzymes. And D, degradative enzymes. So 7 functions of plasmid. Name is in plasmid. P for pily, L for light UV resistant, A for antibiotic resistant, S for secrete endotoxins, M for metal resistant, I for nitrogen fixation, and D for degradative enzymes. Now few more differences between pro and eukaryotes. Now prokaryotes have cytoplasmic streaming absent. Cytoplasmic streaming means, wait, suppose this is a cell. Suppose this is a cell. Now they will stream along the cytoplasm, means flow along, glide through in this motion. It will move from one place to another, slowly, the slow moving. This is absent. This is absent, but the cytoplasmic streaming is present in eukaryotes like advanced features. They are present. This is primitive, not present, advanced feature present. Pinocytosis, absent. Here it's present. Pinocytosis stands for, wait, let me write. Pinocytosis means cell drinking. And another is membrane bound. Both are membrane bound. This same for both. Intracellular organelle is absent. Here it is present, like mitochondria, etc. etc. It is 70s ribosome, it is 80s ribosome. We already studied. This is very important. Cell wall, peptidoglycan for bacteria. And here sterols are present. But in case of fungi, we know it is peptidoglycan and cell membrane has sterol. That sterol is ergosterol. The parasite that has a sterol is Leishmania donovani. Now, only exception is in mycoplasma. It is made up of ergosterol. This is very important. Very important. A question come. This is exception. Mycoplasma doesn't have peptidoglycan. Instead, it has ergosterol, and mycoplasma is the smallest. Okay. So for the introduction part, it was till this. Hope you enjoyed. Thank you for watching. We'll continue it.